seen me speak before. I think some of the slides I have borrowed from previous presentations, but hopefully there's enough new material here that you'll still find maybe interesting. Yeah, if anybody has questions during the presentation, I don't mind if you interrupt and ask. So feel free to. I think it's a little more exciting for me to present if somebody asks questions instead of me just going through all the slides. Um, so I shared my slides with you guys on the chat box just in case you wanted to follow along or at least go through and decide if you wanted to stay for the whole thing or not. So let me go ahead and start sharing. Yes, so today I'll talk about the idea of inverting protein structure prediction models to solve various problems in biology. And the roadmap I will follow is I will first sort of introduce the protein folding problem, how evolutionary information can be used to solve this problem, talking about protein hallucination, and then introduce some interesting ideas towards like how does this help with the protein design problem, or another way to think about it is like inverse protein folding problem. And finally, towards the end, I'll talk about some interesting stuff we've been doing to see, can we actually use these models to predict structures from single sequence when you don't actually have any evolutionary information. So I guess we all know how things happen inside the cell. We hope go from DNA to RNA to amino acid sequence, and that folds up into a three structure. But one of the big problems that we've been thinking about for the last 50 years or so is, can we do the same thing inside a computer? Like, can we go from a sequence to structure? And we know from work done by Kendall and other people in terms of protein folding, and before that, Affenstein and Leventhal, is that the idea of protein folding, you could sort of summarize it as sort of this search in this landscape. So if we think about the protein, there's one global optima and there's a landscape, and this landscape is smooth. It almost doesn't matter where we start. If we can sort of move around this landscape and we find the global optima, then that's going to be our structure. But the challenges here is twofold. One is like we need to somehow define this energy inside a computer. And then two, we need to figure out how do we actually move in this space. And so you might imagine we might be able to use molecular dynamics, but unfortunately with molecular dynamics, it's extremely slow. It requires large computers to actually fold proteins. I mean, you could do it like maybe small conformations, maybe very small proteins, but to do this inside the computer is very, very expensive. But also there might be things that are missing in the energy functions preventing us doing it very accurately. So for some of these slides, it might be a little introductory because I think I was initially preparing these talks for maybe people that don't always work on proteins. But the way I like to think about proteins is a string of beads, and you have sort of hydrophobic beads and hydrophilic beads. And you imagine if you take this beads and drop it into like a bucket of water, what ends up happening is that as you probably would expect, is that the oils come together and the water comes out the outside. And so this property is what we like to call solvation, also referred to as hydrophobic collapse. And you can imagine this could be something that could be encoded or coded somehow in the score function or this energy function that I talked about. And if you actually look at real proteins, we see like soluble proteins, they had hydrophobic residues inside, and then proteins that are actually inserted in the membrane, interestingly, have almost the opposite effect, where you have hydrophobics on the surface interacting with the lipids, and the hydrophilic residues are actually now found just outside of the membrane. But besides these kind of things, you can imagine you might have other energy functions. Like, for example, you might think about distances between atoms. If they're too close, that's not good. If they're far away, then there's no effect. There might be some preferences for side chains. So for example, like this tyrosine, it might have sort of like these three preferences. And so that also helps with the sampling, because instead of doing some molecular dynamics and checking to see the probability, you could sort of predefine some kind of knowledge base potential that favors these conformations. Same thing with the backbone dihedrals. You might have preferences for different dihedrals. And then you can also think about different kinds of bonds, hydrogen bonds and ionic and disulfide bonds. And so if you have all these sort of functions defined, you might imagine then you might be able to go back and actually do this. Like you have this energy landscape, and then if you move around, maybe you'll find the solution. And these are kind of problems that people have been working on for a long time. So like, one is, can we somehow think of a faster approximation that also incorporates some knowledge information to potentially help you get at sort of some of these energies faster? And then the other part is like, how do you actually then build a structure from all this information? And one of the classic examples is Rosetta, which uses fragment insertion. The idea is like, if you could predict fragments for every single protein, you could recombine these fragments and try to predict the structure. And this work was initially led by a group in David Baker's group, but then there have been also other people that did similar things in other groups as well. But one thing about these methods is that it turns out it works, but it's somewhat limited. It works for very small proteins, like less than 100, and also works well when the loops are very, very short. So if the loops are very, very long, you can imagine if you try to recombine fragments, you might never come back around to recombine it on the other side. So these methods work, but they're very limited to very idealized proteins and small proteins. But as we all know, in biology, proteins tend to be very large, often form gigantic complexes. And so even if we sort of come up with the best possible energy function, sampling these structures becomes very intractable. And so the question is, can we somehow cheat? And this is something that I worked on as a graduate student, is we're thinking like, can we somehow use evolutionary information to help us? And one idea here is, imagine if you have a protein string, this, let's say, comes from a human, and these are just amino acids, just a cartoon here. And if we just have a single sequence, maybe we can use some of our physics or chemistry that we know to infer, like, oh, this kind of looks like it can pair with this guy. This looks like it can kind of pair with this one. But one thing is, if you have many, many sequences of the same gene coming from many different organisms, you might be able to do something slightly better. In this case, you might look at these positions and you say, well, these positions appear to be covariant with each other. This position is constant. This position appears to be completely random. And so positions that are constant might give you some information about, is it on the surface or is it inside the protein? Positions that appear random could tell you a little bit about information about like for example, a lot of times these might be actually on the surface, for example, if it doesn't really matter what that amino acid is. And because this stuff is so important, you can imagine taking this string now and try to predict a structure that matches all these covariance patterns. So like this case, the human sequence and the cat sequence. And because this is so super important, you might imagine these contacts are going to be preserved by nature, and then they will be encoded in the multiple sequence alignment. And I mean, there's a lot of people that worked in this area, and I've come up with a little link here that sort of summarizes. If you go to bit.ly 3MR8351, I sort of have like references of a lot of people that worked in this area. I think before I would sometimes write out all the references, but every single time I give a talk, some angry person comes to me, he's like, oh, you forgot to reference me. So I was like, okay, I'm just going to make a website and put all the references there. And then, but if I forgot to mention you, just tell me and I'll add you to the list. Okay, yeah, but this area, a lot of people have been working on, like, how do you extract covariance patterns from multiple sequence alignment and how to use them as constraints in modeling. For those of you who haven't really thought about proteins and protein contact maps, I just a few introductory slides. So here's a protein structure. I'm coloring it blue to red, N to C terminus. And one way to represent this protein structure is imagine like go from N to C, N to C. And let's say if I wanted to show like how the cyan part interacts with this orange part, I can go to the cyan part, go to the orange part, and you can show the contacts here. And so here you can see sort of like here are the helices of the protein. And then the parts that don't have like these large contacts nearby, those are the sheets. And you can see, okay, this sheet is interacting maybe with this loop here. The reason why this is kind of useful is because imagine if you now are able to predict these contacts. We go back, like if you can predict these contacts, if it's just a few residues, you can just show them as lines like this. But when you're looking at a lot of contacts that you're predicting, it's useful to show it as a contact map. And so in this case, these in gray are the crystal contacts just based on some distance threshold. And in blue, what you see are all the covariant residues that we see from a multiple sequence alignment. And these guys are shown as white lines here as well. And so you can imagine if you didn't know what this protein looked like, it was just some really long string, and you put a bunch of rubber bands between the positions, you might be able to sort of bring it all together into a 3D structure. But there are some problems sometimes. And so usually like what we assume is when we see a string like this, and let's say this position is covariant with this one's covariant with this one, our first thought is like, okay, it forms some kind of monomeric structure and all these residues are satisfied in one structure. But what sometimes happens is maybe this protein is a homodimer and maybe these contacts are not formed in one hand, or maybe there's like a two-hand clap where you bring in two hands together And these contacts are formed at the interface of the two proteins. And then you might imagine there's scenarios where there's contacts that are mediated by ligands. There might be also scenarios where different contacts are formed at different conformational states. And so all of these things, we don't consider them to be noise, but they interfere from our ability to predict the structure from a single protein. And on top of that, of course, then there's also noise because of limited data. And for that reason, like if we go back to the example I showed, like there's like this one false positive contact. Imagine like if there was even one wrong contact from here to here, you might get the wrong structure. And so there's been a lot of work done to try to figure out how do you get around this problem. And there's been a lot of work like from like Willie Taylor, for example, doing like fold enumeration, trying different combination of restraints, like you may have heard of DCA fold or EV fold, or sampling lots of structures from fragments like fragfold and Rosetta. And so it's trying to figure out a structure that makes as many contacts as possible. And that sort of leads to sort of a cleaner contact map, as you will. Since then, there's been also a lot of work done to try to filter these contacts. People like David Jones, Jimbo Hugh, been thinking of, can we somehow learn neural networks to filter contacts? And some of these newer methods like AlphaFold 1, for example, or TR Rosetta, effectively do this. They try to filter the contacts. And once you have a filter of these contacts, then you can imagine a very, very simple procedure that just puts rubber bands everywhere and just snaps things in place. Same thing if you're interested, the references for these methods are here. And so just to give you sort of a rough pipeline of how structures were predicted before AlphaFold. So the traditional method is you have a multiple sequence, you have a single sequence, you search against a database of sequences, you construct a multiple sequence alignment. Then you would take this multiple sequence alignment and you'll look at the conservation at every position. So for example, this guy's always green, these guys are blue or yellow, these guys are random. And then you construct a PSSM, like a position-specific scoring matrix, just probabilities at each position. And then you pass that to a neural network, secondary structure. And then based on secondary structure, you select fragments. So for each part of the protein, you try to predict fragments. And then you can recombine these fragments to create the structure. So exactly like the video I showed earlier. You might wonder, like, why is it necessary to do this? And why do people sometimes use PSSMs? And I think this is actually illustrated really nicely by this slide by Nick Grishin's group, where here what they found was if you just scan the whole PDB, often you can find the exact same sequence. So this sequence and this sequence are exactly the same, but the difference is in this one, it makes a beta sheet, and in this protein, it actually makes a helix. And if you take the sequence and you just pass it to CyPred, like one of these secondary structural predictor methods, just using a single sequence, it incorrectly predicts both of them actually form sheets, when in reality, this one forms a sheet, and in this context, it actually forms a helix. But what happens really interestingly is if you actually try to get a whole bunch of sequences, from many different homologs, and construct the profile, like a PSSM profile, probabilities of each position, then it's actually able to correct, like these neural networks can then predict, okay, this is a sheet and this is a helix. And so this is why it's kind of becomes really important to construct these PSSMs, because they actually then help you decide what the secondary structure is. And the reason why we think this is true is because you can imagine if you're in this environment, you will have different preferences for amino acids. And if you're in this environment, you'll have other preferences just based on your neighbors. And so it turns out PSSM is actually very, very powerful, even without thinking about coevolution. Just having a PSSM is already telling you a lot about your context, like who are your neighbors, and based on your neighbors, you might be able to predict the secondary structure. And these are sort of the advancements made like in the 90s and so on, is like having this ability to predict secondary structure. And then once you predict the secondary structure, then you can get fragments and recombine them to some extent. But since then, the other thing that changed, as I mentioned, is this idea is like you take a multiple sequence alignment, pass it to some kind of model, like a Markov random field or a POTS model, get contacts. And once you have contacts, that also makes the problem much easier, because now suddenly you have some restraints that tell you, okay, this side and this side should come together. And so then you could predict the structure based on that information. But now you might wonder, like, where does this AlphaFold come into play? And so what AlphaFold did was they said, well, instead of using these contacts directly, why don't we just take these features directly from our Markov random field or POTS model and pass it to some ResNet, and then have the ResNet predict distances, so the distances between every pair of residues, and then also the dihedrals in the backbone. And once you have that information, then you can just pass that to a minimizer, and get the structure without having to do all this fragment recombination and so on. And it worked out really well, but sometimes it didn't give the best structures, because you can imagine the problem with distances is that you could still have the same distance of atoms like this or atoms like this. And so what actually helps a lot is to say, well, instead of just predicting distances, can we do a step further? Maybe also predict, for example, let's predict this dihedral, maybe predict this angle, maybe predict this dihedral. And so in TR Rosetta, which is one of the methods I've been involved in, is to say, well, instead of just predicting distances, can we also predict all these angles and dihedrals, so the orientation between every pair of residues? And the reason why this becomes quite interesting is because once you have all this information, that tells you all the information you need to reconstruct the backbone exactly. And so if you pass it to a minimizer, even without any additional energy function, you can get back the exact structure. Of course, the sidechain, you still need some other function to get right, but once you have this information, you can get back the backbone. Okay, maybe I'll stop here to see. Does anybody have any questions or concerns before I keep going on?
Everything's okay here. You can keep going on.
Okay. All right. So this is the improvements. Like, since AlphaFold 1 is to say, well, we could also do a little bit step further and predict a little bit more information between every pair of residues. But one thing that happened is, once this model came out, one thing we were kind of curious about is like, well, we know that you need a sequence. We know you need a multiple sequence time. We know you need a PSM to predict these structures really accurately. But like, what if you actually just eliminate all this information? Like, just delete all this and only pass the single sequence? Like, what will happen? And if you do that with natural proteins, like proteins that are found in the PDB, you get pretty bad accuracy. So what I'm showing here is a number between 0 and 1 TM score. 0.5 means you got the right fold, and less than 0.5 is you got the wrong fold. And with natural proteins, we see that the method is not able to predict the structures really well. But one thing that was really surprising is if you actually take proteins that were designed de novo, so these are proteins where somebody sat down and thought about, okay, I have some motif that's very important. I have secondary structures. I'm going to put all the secondary structures. I'm going to put on the loops, and then I'm going to put all the side chains based on Rosetta, and then design this protein. And there's been a lot of proteins that have been designed this way. And it turns out if you take all these proteins and try to predict them, turns out methods like TR Rosetta is able to predict them all correctly. Like, for example, this is a top 7, one of the first de novo designed protein. From just a single sequence, it can actually predict it. And why that's true is still a bit of a puzzle. Like, we're still trying to understand why that's true. Like, some hypotheses are like, well, maybe de novo proteins are more stable. You only need a single sequence, and all the natural proteins are kind of noisy. Other hypotheses is that maybe there's something interesting about, like, maybe somehow all the rules, or the ideal rules, these models have already learned, and they're sort of being recapitulated to some extent when you give it a single sequence. But if you guys have any other ideas, like why natural proteins require more, like an MSA, and de novo proteins have only require a single sequence, I'd be happy to hear about it. But it's still kind of interesting. But that being said, like, we were wondering, well, if it could predict de novo proteins so well, could that mean that we could just use this method to make more de novo proteins? So imagine a scenario where you have a structure, and now you actually want to go reverse and say, okay, I have a structure that I want. Let me pass through the network and try to figure out a sequence that actually will code for the structure. Or you could even take it a step further, say, well, maybe I could just try to make up some new protein that the model likes. And this idea has been around for a very long time. There's a link here about inceptionism, from where people try to understand, like, what are my models learning? So here's an example where you can start from random noise, and then you tell the model, okay, I want a banana. And then you just keep changing this noise until the model thinks that it sees a banana, and then you can look at it, and then hopefully you'll see a banana in this picture. Same thing with starfish or parachute. You might end up finding something weird, but in reality, you can sort of get a sense of what is the model learning by doing this kind of procedure. Another idea is you could start with some random image, like in this case, clouds. And then you ask a model that was trained to classify images, like, what do you see? And let's say if the model sees, oh, I think I see like a fish here, or I think I see like an elephant here. You could tell the model, okay, just keep changing the image until you're very, very confident about what you think you see. And if you do that, you start to see really interesting patterns start to emerge, like a lot of like birds, and I'm not even sure what these are, like cat, a tower, like all these start to appear. And so in some ways, so this is what we refer to as hallucination, is that we can start with some random sequence, pass it to TR Rosetta, and get back some blurry looking image. And then what we could say, well, keep changing the sequence over and over and over, just keep adding random mutations until this image becomes less blurry. And so this is an example. What we did was we took an image and we just kept changing it. It became very, very clear. Similar to how you look at the clouds and you said, keep changing it until it looks like something. And then this could result in a protein structure. And so Ivan Anishinka and experimental collaborators, Sam and Temuka, took these proteins that we hallucinated with using this approach, and then picked a few examples of them to express. And then we crystallized these proteins. And we found that often the case that so this is the hallucinated protein, this is the NMR structure, hallucinated protein, crystal structure. In some cases, our dreams don't always come true exactly the way we dreamed them. So like in this case, here's the hallucination, the crystal structure. There's a bit of an offset between the two sheets. So I think, but still, like a lot of the structure looks very similar. And so in some ways, we're pretty excited. Like even though this model seems to not do so well on predicting natural proteins from single sequence, it somehow learned all the rules of de novo design proteins. And so that just means we can make more de novo design proteins. But there is some limitations, because you might imagine if you can only predict de novo design proteins, maybe if you want to now start to hallucinate some natural functional proteins, it might have a hard time doing that. And so this is something that we're still exploring. Like, can we go and now start to make functional things instead of just looking at proteins that look good? Okay, so besides this idea of sort of saying, okay, let's just start and make something sharp until it sort of matches anything and just hallucinate things, you could imagine you could do something a bit more constrained, where you could say, well, I'm going to start with some random sequence. I'm going to predict something that looks fuzzy. Then let's say I have a structure and I know what I want. And then I could design a loss function that minimizes the difference between what I want and don't, and what I predict. And you could just keep looping this over and over and over until it matches. And so the idea is, like, instead of just hallucinating things, you could actually optimize for a specific category, or it could be some combination of things. Like you could say, okay, part of the protein I want to match something I know or want. Part of the protein could be just free to hallucinate anything. And so we have some ongoing work with this kind of approaches. Now, theoretically speaking, you might wonder, like, why are we doing this? Like, why not just train a model that takes a structure and returns a sequence? Like, why are we taking a model that takes a sequence and returns a structure and then inverting it? Like, what is the theory behind it? And so what we like to think is that these kind of models, they do something, address a very fundamental thing about what we like to think of inverse folding. And so when you design a protein, what you're doing is you're actually designing a conformational landscape. I think a lot of times when people think about protein design, they're like, oh, I'm designing a sequence that has this structure. Like my goal is to go from this sequence, find a structure. But in reality, what you're doing is you're designing this landscape, because what you want to do is you want to find an amino acid sequence that folds into this structure, has the lowest free energy, but at the same time does not fold into anything else. But the other thing you might imagine is if you're not careful, it might have the lowest free energy, but then it's not accessible because you have some kind of barrier. And so nature has been optimizing the sequence to have a very nice landscape, but that doesn't mean that every single sequence will have that landscape. What you need to do is you need to come up with effectively, you need to design a landscape so that your protein will actually fold into that desired structure. And so what we like to think is like traditional methods like this, where you say, okay, I have a method that predicts the sequence given structure. You have this problem where you're only looking at the target structure and you're thinking, okay, I'm going to minimize this energy. My structure will fold into the structure, but then it may actually fold into another structure with similar free energy. And maybe in some cases that might be a desired thing, like maybe you want like conformational change or something like that. But what we think about models like this, when you invert them, is that you're seeing the whole thing. Like this eye is looking at the whole landscape. And so when we finally come up with the sequence that folds into the structure, it does not fold into any other structures. But hopefully maybe the landscape would be somewhat smooth. Of course, that's not really guaranteed because the model is not explicitly looking at the whole landscape, but we're kind of hoping that by minimizing this state but and maximizing all the other states, that maybe that's what we're going to get at. Anybody have any questions or concerns about this hypothesis?
Perfect from here.
I'll ask a question. I guess, so you're saying that when designing a protein, there would actually be sort of penalties for the incorrect conformation? Like there would be sequences that would penalize the wrong fold?
Yes. So that's what we're kind of hoping. So what happens, let's say if you train a model that does this task, like predicts a sequence given structure, the problem is even if you optimize this function, like the assumption is the probability of structure is like 1. Like you think that that's going to be always the right structure, but in reality, there might be a different structure. And so then by optimizing this instead, what we think is happening is that we're automatically making sure that it only folds into the structure, but nowhere else. Now, whether or not this is true, we're still investigating. We wrote up a little preprint about, now published paper on this idea, if you want to take a look at it more carefully. And we do see, like, for example, if you do this kind of optimization, then if you do actual folding inside a computer, that more often than not, the global energy will have the best structure and not have alternative structures. And so this is why we think having this kind of approach to design versus this is going to be better. Any other questions? Okay, I guess I will continue. But if you have more questions,
I have a question here.
Mm-hmm.
So I was wondering here, can you consider this as exploring the sequence landscape instead of exploring like conformational landscape?
I think so. Yeah. So maybe, what like for example, at this particular frame here, so this sequence codes for this landscape. Initially, hey, it could fold into anything. Maybe you could think of that as sort of an unstable protein. But then as you're changing the sequence, you've now found a new landscape. And as you keep changing the sequence, you're finding another landscape. And so in some ways, you're right. Yeah, we're exploring the sequence landscape, but every point along that sequence codes for a landscape in itself.
Okay. We can say sequence landscape is much more complicated than.
Exactly. Like you can imagine, like, let's say there might be one sequence that is like flat, and then another landscape might have something like this. Another sequence might have, I don't know, something like this. And so each sequence kind of, I guess the way we like to think about it, each sequence codes for a conformational landscape. And so then let's say if this is your desired structure, then this would be a bad sequence. This would be a bad sequence. This would also be a bad sequence because you're here, because you would fold into here instead. I mean, an optimal sequence would have a landscape that kind of looks like this. Where there's this there. But now imagine there could also be a scenario where that there is, for example, so another thing that we really like to think about is that it's all about accessible, or the kinetics here, because you can also imagine a scenario where there's like this giant barrier or something, right? So like, for example, let's say if you're here, it's going to be really, really hard to pass this barrier. Maybe if you have a chaperone that can help you, and maybe that's how nature sometimes gets around that problem. But in some ways, it's like you're coding for this landscape, and you're hoping that you've come up with a good landscape that most of the proteins will be able to find the solution inside the cell.
All right, thank you.
All right. Okay. I guess I will continue for now. Okay. So far, I told you guys all about this TR Rosetta model that starts from sequence, build a multiple sequence alignment, and predicts these inter-residue orientations, and then they go past to some model like PyRosetta to build up the structure. But how about other models like AlphaFold 2 or Rosetta Fold 2 that you guys have been hearing about recently a lot for the last year and a half? And so the idea behind those models is that they can take another step back a little more. So what I mean by that is, so traditionally we would first predict some POTS model or Markov random field, and then pass it through a ResNet. What methods like AlphaFold and later on Rosetta Fold did was they say, well, let's have a model that directly looks at the MSA. So you can directly look at the MSA, and then the idea is then you could try to predict some initial coordinates, and then you could refine those coordinates. It turned out later on that AlphaFold doesn't actually predict initial coordinates. It just starts at 0, 0, 0, and then just expands the structure from that. But then you could sort of keep recycling these predictions, and also maybe potentially pick some templates and use that as an initial guess. I think one thing, let me see if I can, there's one slide I was going to show, but I forgot to move it up. So the way we like to sort of think about this problem is that the first part of the model is sort of like a graph inference problem. Like you're trying to figure out, okay, who's connected to who? And then the second part of the model is like embedding the graph in 3D space. And then once you have that initial embedding, you could sort of feed it back into the model and sort of refine that graph. And you just keep doing that over and over. So maybe in some ways, this would be like multi-dimensional, sorry, not multi, like in some kind of MDS protocol. You're trying to like sort of embed this graph with maybe some kind of Floyd-Warsaw of filling in the edges. And then you make some guess and you try again and over and over and over. But the cool thing here is, so we think the reason why this helped was because there's sort of two steps to it. Let's step back a little bit. One is instead of having some external method that minimizes the structure, you go completely end to end. But the other part that's very important is instead of trying to take some Markov random field or POTS parameters, you let the model extract that information directly. Like you have a model that directly feeds in all this information. And finally, templates are also used. So you can sort of imagine initializing a graph using a template. And this will become more important later on, as I'll show you in a few more slides later. And so this is just a cartoon picture that I showed you, but this is an actual image from the AlphaFold paper where you can see what's going on here. And so you have your multiple sequence alignment, you have your templates, you're feeding it in, and then you're sort of, and you just keep doing this over and over, and you keep refining the structure. I won't go too much details about this just due to limited time, but I just wanted to show that there is some interesting similarities between, like, say, the AlphaFold architecture and then the Rosetta Fold architecture, where you sort of keep trying to make this graph better and better and better. Same thing, you try to make this graph better and better and better, and then you're also trying to update the sequences themselves, and you just keep iterating this procedure. One question I often get asked is like, how does this AlphaFold and Rosetta Fold compare? I mean, the architectures are very, very similar, but when the papers were initially published, so this is the Rosetta Fold paper and the AlphaFold paper, initially there was some differences between them, but Mink Young and David Baker's group and others like Frank DeMeo, Ivan Anishinka, and Hustis, they've been working really, really hard on trying to catch up. And so at this point, I would say we've gotten pretty close. There's probably like a few more points here. So maybe a few months later, maybe we'll be here, but we'll see what happens. But the method's getting much better to a point where now we're getting pretty close to getting the right answer. But okay, so now we roughly know how the model works, and we could go into details, look at the code, read the paper. But one other way to sort of learn what the models are doing is to break models. Like imagine you've captured this model or an organism inside the computer. How do you actually figure out what it's actually doing? And one way to learn what it's doing is to start to break some of these lines here. And so some things we've tried to do is say, okay, let's turn off the recycling, see what happens. Let's remove the templates, see what happens. Let's remove the PSM, remove the MSA, just keep one single sequence. Like what would happen? And I think that's another way to sort of learn what our model's doing is just by trying to remove some of the stuff that were initially given to the model. And so the very first test we did, the exact same test that I showed earlier with TR Rosetta, is ask the question, okay, what happens if you just give it a single sequence? Because I think when methods like AlphaFold first came out, there was a lot of press saying, hey, they solved the last 50 years of folding. I see a hand. Reza, do you want to ask a question?
Yes, hello.
Okay.
Can you hear me?
Yes.
Okay, so how do you try to understand how the model works by breaking those lines if people assembled all the lines in the first place, right? they knew like recycling was necessary and they put it in the model. And so how come we don't know what recycling is contributing to this process?
Mm-hmm. Oh yeah. I mean, we know all these things are important to get the best possible solution. But one of the questions was like, well, if you remove these things, how much worse does it actually get? And is it actually necessary? And it just turns out like for some proteins, you can get away by removing a lot of edges, and some proteins you do need those edges. So for example, one big question is like, do you need the MSA? And if you just give it a single sequence, you're still giving it an MSA. You're just giving it an MSA with a single sequence. And then you could ask the question like, does it actually understand folding from the like the first principles point of view, or does it somehow using something from the MSA to figure that out? And the reason why it does make sense to remove some of these things sometimes is because when the models were actually trained, there were some families that only had a single sequence. So the model has seen that scenario before. And also, like, for example, there have been scenarios where there are no templates. So model has seen that kind of background. And then also recycling, there have been scenarios where they train with the one recycle, sometimes they train with no recycle, sometimes they train like 10 recycles. So the model themselves have actually seen pretty much all these scenarios. And now, because it's seen all these scenarios, then we reason that it's valid to go through and start to turn off and then now try to apply it to all proteins and see, do all these principles still apply? Okay, so with that in mind, we're like, okay, well, since the model has been trained sometimes on single sequences, let's try and see what happens if you just give it a single sequence, but now apply it for all the proteins, not just the proteins where there's only a single sequence. And if you do that, probably not too surprisingly, so this is the TM score that I talked about before. If you give it the multiple sequence alignment, it does a really, really good job at predicting it. But if you just give it a single sequence, there are some proteins you can predict from single sequence, but majority of them, it has a hard problem of doing that. So this is very similar effect that we saw with TR Rosetta. But now if we try instead give it a de novo design protein, like just the proteins that we talked about earlier, very, very similar scenario. It turns out for de novo design protein, like single sequence that were designed by people, once again, it's able to predict really, really well. It's not perfect, but you can see, so 0.5 is roughly where you'd say is the right fold. So it's able to predict most of the structures just from 0.5. But then there's a few cases where it had a hard time predicting from single sequence. And so we kind of looked at those a little bit more carefully when wondering like, what's going on? Like, why can't it predict this protein? And turns out one of these proteins is very interesting in that it is actually a transmembrane beta barrel that was designed by Anastasia Verbayeva. But the thing is here, it turned out it can't predict it from just the default 3 recycles. So AlphaFold by default does just 3 recycles. But if you just increase these recycles and you just let it run for as many steps as possible, it actually, after about 15 or so recycles, it's actually then able to predict the structure correctly. So even though the model was trained with fewer recycles, if you just keep running these recycles for further and further, eventually it's able to figure out the right structure. And so we think this is kind of interesting. It sort of tells us that maybe somewhere in the model, there is this graph refinement going on. Like you made some initial graph, maybe based on the single sequence, you have some initial coordinates, you refine these coordinates and you just keep passing it through and through, and eventually you might get the right structure. Here I'm just showing the structure colored by N to C, blue to red. Another way to look at the same protein is to say, well, let's color it by the confidence. So one other really cool thing about methods like AlphaFold is that it provides some kind of confidence for every single position. So you can, here I'm coloring it blue, meaning very confident, red being not confident. And these numbers here are just showing the average confidence. The other really cool thing that comes out of AlphaFold is this predicted alignment error. So for every pair of residues, it also returns a confidence for every pair of residues. But then also it predicts sort of context as well. And this becomes kind of important to see because initially you can see at the very beginning, it has no idea who should be paired with who. The probabilities are kind of equal across the whole board. But then as it iterates, eventually it settles on one graph and it becomes really, really confident about that graph. The reason I'm spending some time on this slide is because you can imagine now, if it has this ability to say, are you good or are you wrong structure, we might be able to sort of repurpose this for lots of interesting applications, because you could think of this PLDDT as sort of being like a site-wise energy function or log probabilities. And you could then also think for every pair of residues is going to sort of being like almost like a pairwise potential to some extent. And so this kind of stuff comes out of the model. Is like, besides just the structure, you get a lot of rich information from the model as well. Any more questions? Just checking. Okay, so I guess I just told you that single sequence is not doing so well. But then we imagine like, okay, what if we just give it a PSSM instead? So like what we're going to do is we're going to, instead of giving it MSA, we're just going to give it like the average sequence and pass it to the model. And so based on some slides I showed before, we say, well, PSSM is very, very important because you can get sort of information about the, at every single site, like what's the neighborhood. And so here, probably not too surprising, we see is that it actually improves a bit. And so maybe what I should have showed is put these two slides side by side so you can sort of see, and then switch between them really, really fast. So the orange is using just a single sequence, and then the red is using a PSSM. So if you just have a single sequence, it has a hard time predicting it. If you give it a PSSM, now there's a bunch of structures that it actually can predict correctly. It still hasn't solved the problem completely, but then if you use an MSA, that would be the green line. It's able to then predict the structures perfectly. And now finally, for the last 10 or so minutes that I have left, I'm going to tell you a little bit of some work that we've done recently with an undergrad in my lab, James Rooney. And we were asking the question, okay, what if we don't give it an MSA and we only give it a single sequence and a template? Will it be able to predict the structure? So no MSA at all, just sequence and template. And what we find is that it's actually able to predict it correctly. So what's happening here is the distribution here. This is the original plot I showed, an original plot with single sequence. You're not able to predict the structure correctly, but if you give it the template, which is the correct structure, it's able to predict it exactly, which is probably not too surprising because you can imagine it could just steal those coordinates and copy them over. But one thing that was interesting though, was that besides just predicting the structure correctly, it actually knew that it predicted correctly. So like when it failed, for example, this is a failed prediction, no structure, the confidence, this is the predicted TM score, is 22. And when you give it the correct structure, the predicted confidence was 84. In fact, if you take this a step further and generate a whole bunch of structures, a bunch of decoy structures, and pass them all as templates to the model, it's actually able to correctly say, okay, these are the bad structure, these are the good structures. Just to give you an example, so no decoy, it's very low confidence, and the predicted structure is really bad. If you give it a decoy that's like somewhere in the middle here, it's only semi-confident about the structure, and then the structure is also semi-correct. And if you give it the right structure, it's very confident and the structure is correct. So it turns out that using this thing that I just showed you about a little earlier, just looking at the confidence at each position and confidence at every pair of positions, you can use that effectively as almost like an energy function to score structures. And so now the question is like, okay, we have some way to extract like an energy function from AlphaFold. How does that compare to, let's say, the Rosetta energy function? And how does that compare to some other models that were trained specifically for this task of being able to just sort of discriminate between structures? And so the cool thing is, it turns out that the method AlphaFold, it actually does a very good job at ranking. And so this is the Spearman rank correlation so just quantifying how correlated this line here is across many, many different proteins. And what we find is that it does a much better job than even Rosetta at the same task. And so this got us very excited, because what that means is that maybe now we can substitute the Rosetta energy function with effectively AlphaFold's energy function. And so, but this also leads to an interesting hypothesis about how methods like AlphaFold and RosettaFold actually work. So our current working hypothesis is that what's happening is that the MSA sort of provides some initial intermediate graph, like that may be slightly inaccurate. And you can imagine like you're almost stepping here. And then the rest of the model just then takes that graph and refines it and brings it down to here. Now, if that's true, you might also imagine if you start here, it might walk to here. And if you start here, you might walk to here. Maybe if you start here, you walk to here. And so this is something we actually test. Like if we just take a bunch of structures that were predicted by people who try to make predictions in CASP. So these are the input accuracies. And then if we just pass them the model and then check out the output accuracies, you can see that it consistently refines the structures. So if you give it this structure, it'll give you back this structure. If you give it back this structure, it'll give you back this structure. And so our hypothesis is that the MSA and the template is some kind of, it's in some ways it's solving the global search thing, assuming that you're close enough, and then it's able to refine it a little bit further. But if you don't have the right structure, I mean, don't have a initial correct structure, then the best it can do is maybe go from here to here. But if you start with something close by, then you can go from here to here. So the MSA is providing a very good solution for the global starting point, I guess you could claim, or at least what we're claiming. Does anybody have questions about this part or concerns?
I have a question about the template. What is the template? Is it polyalanine? Like, is it an alanine structure, or I guess, what is that template?
That's a really good question. Sorry, I should have brought this up. So within the method of, actually, let's step back a little bit. Within the method of AlphaFold, what happens is the template is actually provided as a distance matrix. So what you compute the distances between all the C beta coordinates, and then you bin those up into like from 1 to 2, 2 to 3, and so on. And that's what's actually provided as a template. In our case, our template is just our correct structure or some decoy structure that we've generated with some external method. Yes, so to answer your question, the template is actually not a full structure. It's just a histogram of the C beta atoms, which I think is actually pretty exciting because methods like, for example, so for example, method like Rosetta, in order to score the structure, you need to have the full backbone and all the side chains. So you need all that information. With AlphaFold, you literally just give it just the backbone information, even more so just the C beta information, and everything else you don't need. And it's still able to score that structure just from that information.
Thank you.
Mm-hmm. Okay, so I think I just have a few more slides left, and I can stay for longer if you guys have questions. But now, so this made us kind of excited because we're thinking like, okay, we can take AlphaFold and give it some random sequence and it will give us a structure. Like this is what we've been using for hallucination and design. And we could take a structure and give it to another method, another AlphaFold instance, and use it as a template. And in this case, we can give it the right sequence. So in some ways, we now shown that you can use AlphaFold as an energy function. And we also shown you can sort of use AlphaFold and TR Rosetta as effectively a structure generator. So the question is, what if we just link these two models together and just keep cycling this over and over and over? Well, we have eventually arrived at the correct structure. So there's a lot of stuff going on in this slide. So let me show you a slightly simplified version of that. So imagine we start with some sequence, which is like our correct sequence. We mutate it, and we get back some histogram. And we take this histogram, we feed it to another method, another AlphaFold instance. And in this case, we give it the correct sequence. And as I showed before, that you can then use the method's confidence metric to sort of select which is the right structure. And so then if you just keep cycling this, will you eventually come up with some kind of mutant that gives you some interesting structure that this method thinks is correct. Okay, so this is the setup that we've set up. And here's, and here's the animation showing this going in action. So what I'm showing here is the TM score of the structure that is sampled from this generator, the loss function that we're optimizing, which is the confidence. And here I'm also coloring by confidence. And then the structure also colored by confidence. And here I'm just randomly changing that sequence via gradient descent. And what I'm trying to do is I'm trying to find a structure that AlphaFold thinks fits that sequence really, really well. And the cool thing is it actually sometimes works. Like you could sometimes start from the wrong structure and you can just keep changing that structure until AlphaFold becomes really, really confident and it actually ends up being the correct structure. Here I kind of cherry picked one example that looked really good, but in reality, what often this is what happens often. Like sometimes you find this alternative minima and you kind of get stuck. Some cases you find the solution. Some cases you're just stuck sampling really globally. You never find the right solution. And it's actually a mixture of all these different kind of things. But the cool thing is often than not, you do see some kind of how do you say, getting close to the right answer. And here's a summary slide for that. So this is the default method predicting structure from single sequence. And this is after this optimization thing I just showed you. And what you can see is often it does a really good job at predicting the structure, but sometimes it gets stuck. And one thing that's probably kind of interesting, if you kind of stare at the slide closer, is that if you are roughly maybe initially predicting a structure, maybe at 0.3 TM score, you can often make it better. But if your structure is so far away, you're kind of stuck in this thing. So like if your initial prediction is not so good, if you do this optimization, you kind of get stuck in some local minima, which is sort of true of a lot of structure sampling protocols. So one thing we're currently trying is saying, well, can we start from many, many different starting points? Because right now, the only thing we tried is like, hey, let's just start with the wild type sequence. But you can imagine starting from many, many different starting points, leverage a whole bunch of computers and do a whole bunch of sampling. And maybe what you will find is there's all alternative minimas and this fails, or maybe if you sample enough, you might find actually this solution here. And so this is sort of ongoing work as we're trying to see, can we now actually use AlphaFold as both a generator and a discriminator, link them together to now maybe start predicting structures from single sequence. And a few other things which I don't really have enough time to talk about today, but the other thing we've been playing around with saying, well, we can use the confidence, like we can use the confidence to refine the multiple sequence alignment. So for example, you can say, I'm going to start with my original MSA and maybe my prediction is not so good. And I'm going to just keep changing my MSA and then until the confidence gets much better. And often that results in a structure that's actually more correct. Other things you can mention is you can, let's say if you have a structure of a protein and you want to design a binder, you can just keep optimizing the confidence. Like say, I'm going to keep changing the sequence of my binder sequence until the confidence between the binder and their target becomes very, very confident. And then maybe that's a way to sort of start designing interactions. And so I guess the big picture is like, we think we sort of unlocked the energy function of AlphaFold. And now this energy function could be used to like one, sample structures and maybe solve the single sequence structure prediction problem. Or two, optimize the MSA that actually goes into the algorithm. Like say, maybe we can get a better alignment. Or three, maybe start to now to design proteins with these kind of functions by just looking at the confidence and maximizing those confidence metrics. And I guess with that, I'll say thank you. A lot of people in my group that I didn't get a chance to talk about all their work. We're always looking for collaborators and people to work with us. So if you're interested, feel free to reach out to me. Also, I just wanted to bring up that I talked a lot about stuff that we did, but in reality, it's a huge community. There's a lot of people that are actually doing very, very cool work that is very similar to ours. So for example, David Jones has been also trying to invert structure prediction models for protein design. And other people have been working on saying, well, let's design a protein from a model that predicts a sequence given structure or some kind of general description. So there's a lot of stuff going on, and I'm not the only one doing this stuff. So I included these links here. And finally, just a shout out to CollabFold. This is an algorithm that, or I guess a few of us got together and we tried to make CollabFold accessible for people. And I don't have time to talk about it today, but a lot of people involved in that. And here's a link towards that. All right, well, with that, I think I'll end it. Thank you very much.
Okay, great. Thank you very much, Sergey. And now we are open to questions and commentaries. People that is in Zoom, they can raise hands, for example. Well, here we have a question from Matthias.
Okay, so first of all, thanks, Sergey, for this wonderful talk. It was really interesting, inspiring. I do have a couple of questions, actually many. I will try to ask you a few of them and then allow the other people to ask their questions. The first is this idea of using, as the single prediction to get generate a template in order to feed AlphaFold for your actual query. So the thing is that what I'm seeing is that you're first, you need to came with a sort of synthetic sequence, which actually folds, or it's not that it folds, but actually AlphaFold is able to fold very similar to the query system, right? Through query sequence. So is that a problem, or that may be a problem for stacking into this optimization procedure, because you really need to find that sequence, and maybe it is not. I don't know if it's possible or not. So that would be kind of my third question.
No, that's a very good point. Yes, there's definitely going to be scenarios where maybe a vector does not exist that will give you that structure in the first place, right? Yeah, that's a very good point. We are trying to address that right now by purposely making it supervised. Say, okay, let's just purposely make our loss function the correct solution, and then see, is it even possible to find that sequence? And you're right, there are definitely cases that we were not able to find a solution. So some of those did indeed fail because there just does not exist, or at least we're not able to find a single sequence that will give you back that structure. This appears to be more often the case for very large proteins that have like very, very long loops. And so maybe in those cases, just really, really hard to find the sequence that codes for all that information. That's a really good point. The other thing we're thinking about how to address this problem is to think about maybe we can use an external structure generator. Like for example, we could just use Rosetta and generate structures. And that's something we're beginning to look at now. We were kind of hoping that maybe just backprop and somehow magically will solve everything. But it turns out that for the problem like you bring up, like, hey, maybe it's not even possible to find a vector. And two, as with any optimization problems, you do get trapped in local optimas. So you might have to do some kind of a global exploration at the beginning.
Okay, so my second question was, so basically I see your strategy like sort of a threading or homology modeling. Let's say you give a template which is good enough, and AlphaFold is somehow going to fit the sequence through this model. So I'm just thinking in two possible scenarios. One is, why instead of giving like a sequence or a model generated from a sequence, just give we know most of the foldings space, or many of them, so we can actually give different structures, even those which are very distant related, and see what happens. So that could be one possible scenario, which is very similar to Itasar. And the second thing is, well, why not modifying the structure instead of changing the sequence? Because that could be like sort of minimization, rotations, or repacking, or whatever. So have you considered these strategies?
Yeah, the first one is an interesting idea, because you're right. I mean, assuming that every single fold has already been explored by nature, you could just make a list and just go down that list. I think the tricky part there is that AlphaFold didn't learn how to thread. Like, you still have to give it some thread. But maybe if I understood your suggestion is, maybe we can almost try to optimize that threading. It's almost like, can we learn a threading that is more confident? We actually haven't tried that yet, but that seems like an interesting idea to try. It would be to say, okay, let's just get a database of all folds and then just try all possible threads. It might be a little more complicated because you might have to do like insertions and deletions, and that might also be an optimization problem in itself that you can get stuck. But it seems like an interesting thought. As far as your second question, like, why not just directly change the structure? We did actually try something like that. We said, let's just directly optimize the structure. And what we found is that you can actually find adversarial examples. Like, you could start to come up with something that doesn't even look like a protein, and will start to increase the confidence. What happened was AlphaFold was trained with real sequences and also trained with real structures. So it's never been seen like sort of completely disordered or weird looking proteins, things that are not proteins. So when we try just directly optimizing the structure, and just let it like have bond lengths, I don't know, if 20 angstroms and so on, you sometimes see weird things and like you can trick the model into being confident. And so in some ways we need to restrict it to be a real protein. Maybe one way to solve that problem would be to combine AlphaFold's energy function or this unlocked energy function with some other external force field or something like that. And this is something we're now beginning to explore, saying, hey, can we combine it with Rosetta and then sample with Rosetta's fragments, and maybe that could prevent us from going into weird space.
I don't see if there is somebody raising hands here still.
I have more questions, so I can keep going and going.
Still, there is no question from the audience in Zoom, so we continue. And the one in Zoom that wants to make a question, just raise hands and we go to you.
I'll ask a question. I study virus proteins, and I find that a lot of them have predicted structural homology to another thing, but the sequences are nothing alike at all. And often there are a very limited number of homologous sequences to take advantage of the multiple sequence alignment thing. I wonder if there's any way to use like predicted structural homology to make multiple sequence alignments and use that information to feed it in somehow. I don't know if that makes any sense, but
I think it's I mean, yeah, it is a little bit of a chicken egg problem. Like you first have to somewhat predict the structure correctly to make that inference about that it has similar structure. But I mean, if you have some clues that it might be the right structure, I mean, maybe as the original, I'm sorry, I forgot your name, the one who just asked the question earlier. But the idea would be like, well, maybe you could phrase this as a template problem. The idea would be like, well, if you have some hypotheses about that this might be the right structure, maybe we can use this machinery that I just talked about and like enumerate. So the idea is like, instead of trying to come up with a multiple sequence alignment, maybe we could just try a bunch of structures and see, okay, does this viral sequence fit this structure? Does it fit this structure? And so on. So maybe we can make it into a homology search or a threading algorithm to some extent. So that could be kind of fun to try. But I guess to answer the original question is, yeah, if you can't have sequences, it's kind of hard to do it. But I think maybe we can reuse this machinery that you just talked about to maybe solve that problem.
I still have a couple of more questions. The first is, what about, because I really like this idea of, AlphaFold is able to say what is right and what is wrong. So what about trying this score function for docking? Because, sometimes, AlphaFold, if you feed AlphaFold with the two sequence of spreadings, it may came up with a good model, but sometimes it doesn't. So what happens if you just give different templates, let's say of your a protein and a ligand and in different orientations, and let's AlphaFold decide if it is right or wrong.
Yeah, that's a really interesting idea. There was a paper by Dima. Let's see. Let me see if I could find it. I'll post in the chat box here. There was a paper where what Dima did and his group did is they took a few hypotheses. I think they only had like 5 hypotheses, and they tried to re-rank them. So docking conformations, but I don't think they tried like enumerate like the entire docking landscape and then score them. But yeah, I think you're right. You could definitely do the same thing with like a docking conformational landscape and try to score it. Would be interesting to try. So we haven't tried that ourselves, but the closest that I've seen is this paper from here. Maybe I'll share my screen so you guys can see it. So this paper here, they if I remember correctly, they had like 15 models, and then they try to rank these 50 models using PAE values. But they didn't try like score like all possible conformations of the docs, which would be kind of cool to try, I think.
Okay, and another thing is, when you generate this initial random vector, does it has to be random? Because, you know, there is this new preprint about prot GPT-2, which is an AI algorithm that actually is able to sort of speak the pairing language. So it's gonna to say words or phrases which actually mean pairings. So you may try to and those sequences do fall to something that resembles a natural pairing. So what if you fit your algorithms with something which is clever than just a random sequence?
Oh yeah, that's an interesting idea. So one thing, unfortunately, about language models that have been applied to proteins is that they appear to be still limited by the same multiple sequence alignment information. What I mean by that is if you take any of these language models trained on proteins, and if you look at the ones and you ask the question, how accurate are they? Turns out that the proteins that it's most accurate on are proteins that actually have a huge protein family. And then proteins that have just like one or two sequences, the model is really, really bad at making any contact predictions, mutant effect predictions, and so on. So our hypothesis is that language models at the end of the day are just Potts model storage and lookup algorithms. So the idea is that you have a single sequence and you could look up a Potts model, the parameters of the language model. And so the reason I'm saying this is like, I think initially we were pretty excited about language models, but now we're worried that at the end of the day, they're not adding any new information that isn't available from like a multiple sequence alignment. Because they seem to be restricted to large protein families, in which case you often have a really good multiple sequence alignment, and you could predict that structure already. But that being said, there is some evidence that even when you have very low number of sequences, it might be able to borrow information like some kind of statistics from neighboring families. So it might be worth looking at that a little bit, but I think right now I'm kind of worried that it's not really adding any new information that isn't available in multiple sequence alignment.
Okay, so sorry, but I do really have several questions. I don't know if there is some other one in the audience that
not yet. But maybe during the discussion appears somebody that.
Okay, so have you tried this idea of templates or with these metamorphic pairings? Because I've been playing with some family of metamorphic pairings, you know, like so for those that you know, there's some family of metamorphic pairings that during evolution have acquired this property, and some have lost these properties. So in some cases, you can fit the algorithms with sequence of pairings that are known to fall to a particular folding, and or give alphaphold sequences that are known to be metamorphic or fall to the other folding. And it is very interesting that sometimes alphaphold is able really to fold one or the other folding depending on the sequence that you are fitting in. So what I'm just guessing is, or trying to see, what if you give AlphaFold as template each of these possible structures for that query sequence?
Mm-hmm. Yeah, that's an interesting idea. I mean, I think in the AlphaFold paper, in the one that was submitted to proteins, they discussed this idea saying, well, for some GPCR targets, maybe you can try different templates. And I think in both cases, it was pretty confident, but then they had multiple hypotheses. One thing that we tried, not a metamorphic proteins, but there is a data set. Have you heard of GA and GB proteins, where what they did was they took two different structures that are very, very similar in sequence, and they try to find a series of mutations that goes from like one conformation to the other, a series of mutations. And in that data set, we actually do see, like, if you try to score the sequences with one template and score the sequences with the other template, you actually do see like a sigmoidal transition. Like, so the sequences coming from this one prefer the correct structures, and the sequences coming from this one prefer the other structure. Now, the transition itself is not exactly the same where it was predicted, but I think it should be possible, like with, I guess with as any energy function, is to maybe start to think about like maybe some of these guys have multiple states and then score them, but we haven't explicitly looked at metamorphic proteins ourselves. Yeah. But that's definitely a good idea. Mm-hmm. Oh, sorry.
There is a question first from Flavio, and then Rodolfo has raised the hand. Flavio say, have you tried something on orphan genes, meaning natural occurring functional genes with no homology to any other genes?
Oh yeah, that's a good. So this is one of the things we were kind of hoping to try next, but we haven't tried yet. So right now we're just thinking like, okay, we need to make this sampling as powerful as possible. But yeah, one of the hopes is to apply these methods to genes that have no MSA. But it's also probably a good control to do, because one potential criticism to our work would be that, well, even though you're giving a single sequence, it's probably seen that fold before, and maybe it's learned these folds, and you're just recapitulating them. So trying them on real orphan proteins would be a good test to do, which we haven't done that yet. It's a good suggestion.
Rodolfo, you can speak.
Hi, sorry, it's a holiday here. Well, first, Sergey, thank you very much for your talk. I don't know, well, let's see. I know there's people that have been using the Rosetta energy and iterative mutations to improve protein stability. I mean, using the Rosetta energy as a kind of proxy to thermodynamic stability, right? And you showed us now that it appears that the alpha-fold energy works better. Do you expect that alpha-fold energy will also help us in understanding better thermodynamic stability of proteins?
Yeah, that's an interesting question. I think it would, but there is a little bit of a, how do you say, a double-edged sword here, because what happens is AlphaFolds only understands proteins that it can predict. And let's say if you have a natural protein that AlphaFold cannot predict at all, and maybe there's like, doesn't even exist a sequence that can predict it. So I think unfortunately, the applications are somewhat limited to maybe smaller idealized proteins. That being said, we did do some experiments that suggest that maybe you can use this trick to do better sequence design. And what I mean by that is, let's see if I can find a slide I wanted to show. 1 second. Here we go. I think I found it. So one way to sort of answer this question, like does AlphaFold understand, or does AlphaFold understand sequence design better, is to actually just try to design proteins with it. And so the idea would be like, okay, you start with some structure with a sequence, you remove the sequence, you find a new sequence, let's say with AlphaFold, the Rosetta fold, or Rosetta, and then you compare how similar the sequence is to this sequence. And so the idea is, if this method is really, really good, then the sequence recovery will be high. If this method is not so good, the sequence recovery will be really low, like as far as the amino acids and how similar they are to each other. And so the idea would be like, you can start with some structure that you want. You can get a distance matrix from it. You start with a sequence, you pass it to AlphaFold, and you get a prediction. And then you can take the difference between these guys, back propagate through the model to get gradients, and then apply these gradients and just keep looping this over and over and over until these distances and this loss is minimized. But now the other thing you could do now, given our new bit of information, is to say, well, instead of just trying to find a sequence that matches where this loss is minimized, what if we actually then also take the template and feed it in as a template to AlphaFold? Does that actually improve things? This is just an animation showing here how that works. It's like you just keep changing the sequence until you find the structure that matches the desired structure. But the answer is that yes, it does improve. So So this is, if we design a new sequence with AlphaFold using no template information, our sequence recovery is maybe around 25 or so. But if you add the template information, it actually is able then to actually find a sequence that has a higher sequence recovery. So it does look like you might be able to push the model into the right structure space. And then and we think that it pushed in the right space because the sequence recovery also improves. And so this sort of suggests that it could potentially be used as a way to further stabilize proteins. But we have no experimental data yet. This is just showing on the computer, if we just design new sequences and we include the templates, you can get an improvement. We've also did the same thing with another data set, just a CAS data set that was not in the training set. This is free modeling targets, and show the same thing, is that you can see an improvement in sequence recovery. Yeah, so I don't know if that completely answers your question.
Yeah,
but
sure.
It does.
That's
it suggests. Yeah, so it suggests that these kind of things could be helpful. Yeah. Mm-hmm.
There is another raised hand from Martin Graña.
Quick one. Thank you very much for your time and your generosity. For a bunch of Southerners nuts. So, just a curiosity, what on your opinions of future effects? Do you think the amount of reliable structures that we will have at hand will shed some light at some old discussions on, for example, convergence versus divergence of superfolds, let's say? Because besides the bias of all the training sets, including the sequences sampled by evolution and sequences sampled by us, you have, well, the coevolution, the alignments, they are evolution, they reflect evolution, but they also reflect physics. I mean, so do you think, so they are quite intertwined. So do you think, well, if you thought on this, do you think it could shed, because there are all discussion, you still have the physicist who says, okay, a Tim barrel is an easy thing to do, and there's convergence everywhere. And other people said, well, perhaps not much, there's a frozen accident. We started with this bunch of folds, and then this was useful and easy to do, yes, but it's the same as 2 billion years ago. So, well, perhaps it's an important subsidiary question.
Yeah, I mean, I don't know if we'll ever be able to resolve that question exactly, because as you're saying, like, there's a possibility like certain folds have appeared multiple times. But I think one thing it might help resolve is, I think people like Nick Grisham, what they often do is when they're trying to make connections between structures and folds, they try to find the intermediate structures. And what I mean by that is, like, we'll say, okay, maybe this guy and this guy are very, very distant in sequence, but they have the same structure. The question is it convergent evolution or divergent evolution? Well, what if you predict like this structure, and then you also predict this structure, and you also predict this structure, and maybe by sequence they're close. Like, let's say this is within 30%, this is within 30%, this is within 30, and this is it. So even though these guys might have very, very low sequence similarity, maybe all the paths connecting these two have really high sequence similarity. And before, we never had these intermediate structures, but now maybe we can predict these intermediate structures, and then based on that, make some claims about convergent evolution. So I think my suspicion is that that's where, because before, the way like Nick Grisham did it would be, he would like go through the PDB and find these intermediate structures and try to make these connections. But now suddenly we can predict all these guys. That being said, unfortunately, AlphaFold's still kind of limited by having enough MSA, large enough number of sequences. So let's say if you have like an orphan guy here, it doesn't have any sequences around it. Then, so maybe if I draw like a whiteboard, so like the idea is like, let's say we have some structure here, and then we have some structure here, and there's a lot of sequences here. There's a lot of sequences here. There's a lot of sequences here. A lot of sequences here. So, and then maybe there's like only one sequence here. And so maybe there's going to be like we can't connect all the paths. So like say, we take these sequences, we predict this structure, we take these sequences, we predict this structure, and then these sequences are close enough, these sequences are close enough. Maybe there's going to be no path between them. So there might be still missing parts. But maybe this is where we could do the template threading. Like we could take the sequence, thread it on the structure, and then maybe you can actually make that connection. I think this is something really cool to try in the future, but and something that we might be able to do now with these methods.
Good. I need better GPUs or bigger.
Yeah. So
that was another curiosity. Are there any plans to because we are somehow limited. We don't have a strong and big GPUs. You are somehow not somehow. We are limited. Period. And Collab, it's a very we are very forever grateful for thankful for that. But still, we have some issues. Well, when sometimes I end up figuring out. I mean, constructing stuff which is shorter just to make it fit on the size constraints. So, are there any plans to keep on with the altruism for well, or not? Perhaps no.
So I guess the potential good news is that Rosetta Fold has now cut up to the same accuracies of AlphaFold. So what that means is now potentially you can submit a sequence to the Rosetta server, which has no limitations and GPU. It's going to be slower because it's in queue, and then you're going to be waiting for your turn. But I think one of the cool things is that we do have a Rosetta Fold server. I think right now it's still using the old version, but our plan is to update it to the latest version after the CASP. So right now we're testing the new version of Rosetta Fold. By we, I mean actually Mink Young. She's testing the Rosetta Fold server, and then at the end of CASP, we're going to make it a public server. And then you should be able to submit sequence and predict everything. From the DeepMind side, so DeepMind also promised, I believe, by the end of this year to try to predict the structures for all of UniProt. I think UniProt 90, not exactly the whole thing. So over 100 million structures is what they promised to release. So then the hope would be that hopefully your protein of interest is going to be either one of those proteins or at least close enough to one of those proteins, because I think they did it on 90% cutoff. Okay. So I guess to answer your question is Rosetta Fold is a server and should be able to give structures as accurate or better. And that will become public later this year. And then DeepMind will also is planning just to predict everything. And so then that should also potentially eliminate the need to run Collab.
Great.
Yep.
Thank you very much.
Of course.
Yeah, I don't know, maybe there's this technology, the perceivers, which actually are meant to, tackle this issue of memory, because, I mean, the matrix that you have to solve is very huge when you go to very large sequences. So perceivers, I think if they manage to implement this kind of technology, maybe they can somehow improve this memory issue.
Yeah, that's an interesting. So there's actually one thing I didn't talk about, but there's a professor in Columbia, Mohammed Al-Qurashi. He recently re-implemented all of AlphaFold with PyTorch. But one other thing they did was they implemented some of this chunking code that was released recently by Google. So the idea is like these. So where the memory is hit the hardest is constructing this giant attention matrix between all the pair residues. And that's a both an expensive procedure, but it requires a lot of memory to compute. It turns out you can actually break that calculation into little chunks and then run them in serial instead of, and because of that reason, you might run longer, but you might require less amount of memory. And so we haven't yet, but we think by using maybe the OpenFold implementation and then seeing the chunks, maybe you can get away with like a smaller GPU. I mean, it's going to take a longer time to run, but you might be able to get away with it. Actually, let me see if I can find that paper for you that talks about this. I think there's it just came out last year, and I think they've already implemented this procedure in the OpenFold algorithm.
Okay, so I have kind of my last question. is related to, again, this idea of the AlphaFold as an energy function. It's how do you check on the replicates as a measurement of somehow this energy? Because something that we have found is that when you have a structure, let's say of a natural purine, and you optimize this structure for expressing in E. coli or whatever, using for example Rosetta, so you get some mutations which should improve stability. And something that we have observed is that when you make the predictions of this structure, the new optimized structure with AlphaFold, even if this structure was predicted with a very good scoring, I mean, above ground truth, some regions that used to be mainly loops or other part of the protein that used to be, you know, like noisy in terms of the replicates, they improve. So is that meaning that probably, again, it's maybe it's that you are starting from, or you are giving a sequence which is more optimal. So, you know, it's like, is the noise in the cases when, because the problem is that you have like a threshold, right? Because after you reach 100% of your score, then, I mean, what does it mean, the energy, right? So the thing is this noise informative? So maybe the fact that it is easy for AlphaFold find every time a good structure for that sequence, maybe that's also a point and something that's pointing that it's a good energy function.
I see. Yeah, I guess it's a lot you said at the moment, but I think there is definitely some truth to that. I think because if you look at the so one thing that AlphaFold does is it tries to make random mutations to the sequence to make predictions. So one thing happens. So when I talk about these recycling, what happens at every recycle, they try a different MSA and some random mutations to that MSA. So every copy of the MSA, you randomly mutate 15% of the sequence. And so what I'm wondering is if it's possible AlphaFold might be already capturing that information and actually helping with its prediction. This idea that each replicate, you can think of each recycle as being almost like an independent replicate. And that sort of is helping then predict the structure because you sort of introduce some perturbations each time. The other thing I was thinking is in the context of protein design with AlphaFold, one thing we also do is we keep the dropout layers on during design. So what's happening is in the model, what you could do is instead of just running the model once, the same time, what you could do is you could randomly perturb the nodes in the model by just setting them to 0. And I don't know if this is actually going to help eventually like in experiments, but we think by doing that, it might create a more robust sequence. Because you might imagine a scenario like, I think exactly what you were saying earlier is like, as soon as you maximize the confidence, there's no more incentive to change the sequence anymore. But if, let's say, you continually keep adding dropouts and you continue messing with the sequence, you might find a more robust sequence that will no longer be, how do you say, maybe that was a more stable stuff. But right now, we're not really using that in the energy. We're just sort of using that as a sort of auxiliary extra step. All right, well, I think actually, unfortunately for me, I have to run for another meeting in 2 minutes. So if there's any last question, I'm willing to, but also please send me emails if you have any more questions or thoughts or hypotheses. And I'm also really happy to collaborate with people. If you, let's say, if you want to work on some template stuff or anything, I'm happy to discuss. So just send me an email.
Okay, that's great, Sergey. Again, thank you very, very much for your talk and the answering of all the questions.